recall that after conception, the stages that the conceptus went through is it was initially a conceptus, and then it started rapidly splitting as it was bouncing along in the fallopian tubes. And at some point, as it's going through, it just becomes this cluster of cells that we refer to as the morula. Then the morula, which is just a cluster of cells, eventually becomes a hollowed out little ball that we refer to as the blastocyst. And that is about when it would be right here. So notice that it's coming from the uterine tube into the uterus here. And what we are waiting for next is the process of implantation. Implantation is going to be when this blastocyst makes contact with the endometrium in order to burrow inside of it and essentially make contact from fetal tissue to maternal tissue. So this is going to happen between day 4 through 7. That's when the blastocyst actually makes that contact with the endometrium. And this tissue differentiation starts occurring. So there's this union between the two tissues where there's going to be eventually the formation of the placenta, where there's going to be a communication of nutrients and oxygen, and also taking wastes away from this newly developing embryo. So at day 8, the blastocyst digests the uterine wall. So it has to kind of eat its way in there. And the endometrium has an interesting response to that. It's not entirely inviting to the whole process. So the endometrium, the uterus, kind of responds almost like this is an infection in the area. So there's some inflammation that's occurring here. And you can see some blood vessels and some clotting is occurring in the region around here. So at day 12, you can see that the blastocyst is progressively invading that uterine tissue. And then eventually it's going to reach those blood vessels. And reaching those blood vessels that are found in a little closer to the myometrium is going to be important, because it's at those blood vessels that the developing tissue can now form the placenta. So at day 16, the uterine epithelial cells grow around the blastocyst. So this is in essence, the maternal tissue surrounding the blastocyst, and the whole process of implantation has completed. So now that implantation has taken place, now we get the formation of what are called the extraembryonic membranes. So this is essentially the union of the fetal tissue with the maternal tissue that is going to eventually make things like the placenta and the umbilical cord. So where is the embryo in all this? So for all this tissue that you're seeing right here, where is the actual developing new human being? Well, it's this tiny little disc that you see right here. So the formation of a new human being really just starts off as this small little disc. And as we'll see, it's going to have 3 distinct layers. And those 3 distinct layers are eventually going to become the different organ systems that we have. So what I'm pointing to right here is the embryo proper. That is the embryo. That is what's going to become the fetus or the baby. And there's going to be a yolk sac. So here is this yolk sac right here. And what happens in other species is that the yolk sac is going to give nutrients to the developing embryo, but that's not really how it happens in human beings. In human beings, what happens is that as the embryo is developing, a certain amount of that yolk sac kind of goes into the center and becomes the digestive tract. So you can kind of see this yolk sac kind of poking off to the side here, and then eventually here it is off to the side here. All right. We also get the formation of the amnion. So the amnion is this clear fluid that you see, this bluish tint that you see right here. The embryo is going to slowly grow inside of that, and this is going to become the sac that holds the embryo, and it's going to be filled with the amniotic fluid. And then we also have the allantois. The allantois is the tissue that will eventually be the umbilical cord. So you can see it starting right about here where there is that connection, and then you can see the blood vessels penetrating inside there. And then eventually what we're going to have is a communication where there is going to be nutrients that are brought to the embryo, and then wastes that are taken away by that allantois. And then lastly, we have the chorion. The chorion is the most superficial membrane that encloses all these other membranes. So you can see all of that on the outside right here. So this is the chorion right here, and you can see that that is surrounded by blood vessels as well. Now at week 3 to 8, we have reached the embryonic period. And remember that the way that this whole new organism had begun is it was just one single cell. It was one single new cell with 2 pronuclei, one from the mother and one from the father, that came together. It just started as DNA. And then eventually these cells started replicating, and slowly we get the formation of what really is just a disc. So there is this disc that exists that is the embryonic tissue. And there is a formation in the beginning of what is a primitive streak. So this streak that you see going down the middle, it's just a line. We call this the primitive streak, but that primitive streak is very important because that primitive streak is going to define what is right versus left. So before, it was just a bunch of tissue. It was just a cluster of cells. But now with this line that divides down the middle, the right versus left distinction starts giving the whole disc an idea of what is top, what is bottom, what is right, what is left. And that primitive streak is actually going to become what will eventually be the dorsal body cavity. So recall, the dorsal body cavity is what encloses the brain as well as the spinal cord. We also get gastrulation. So there are 3 distinct layers to this embryonic disc. So this process of gastrulation is where we get the formation of these 3 layers. So layer 1, 2, 3. These layers are named the ectoderm, the mesoderm, and the endoderm. And these 3 layers are going to give rise to very specific tissues that you have already learned about all through the course of A&P1 and A&P2. So in weeks 3 through 4, we see this disc that has these 3 distinct layers. And what that disc does is it folds in on itself. So it essentially, it goes from being a disc to being a tube. Now, this diagram is a little bit weird to make out if you don't know what you're looking at here. What you're looking at is the disc from a sagittal view right here. So that's the sagittal plane that you see slicing it there. And then from the transverse view right here. So what the diagram is trying to illustrate to you is that that disc of tissue, here are these 3 distinct layers right here, fold in on themselves and make a tube here. And you can see that in the transverse view a little better. Now, if this doesn't look like what would eventually become a human being, I want you to just think about for a second, for this particular view right here, think about what your body would look like if we were to take a transverse view right through the center. This is where your digestive tract would be. This is where the area where your digestive organs would be found. This is where all the musculature of your back would be. This is where your spinal cord would go through. So you can see that really in just folding this disc of tissue and forming a tube, this will give rise to what eventually will be all of these different tissues and layers that we find on the inside of the body. So once this folding has occurred and kind of all of the components are in place, those 3 distinct layers are going to start forming into organs. And we call that process organogenesis. So these 3 primary germ layers eventually will form all of the major organ systems of the body. So your 3 major germ layers were the ectoderm, mesoderm, and endoderm. So these are the 3 layers of that little disc that was the fetal tissue. And as that folds, you get this process of organogenesis, and you can see that the ectoderm becomes some major structures. You get the nervous system, you get your skin, hair follicles, all those parts of the skin, the major parts of the skin. You get all of your major canals, and then eyes, salivary glands, things like that. So you can see that the ectoderm becomes some more specialized tissue that is involved with the nervous system. The mesoderm ends up becoming your musculoskeletal system, so muscles and skeleton, and then you can see some of the like smooth muscle and fat that would be found like in the bone marrow and kidneys, uterus, etc. All of that comes from the mesoderm. And then the endoderm ends up becoming the like inner linings of certain regions, like your digestive tract and your respiratory system and your bladder. So those like epithelial tissues and such emerge from the endoderm. So from just 3 germ layers, you get the formation of very distinct organ structures, and that is organogenesis. So up to this point, how has the fetal tissue been getting the nutrients that it needs? Well, it's actually from breaking down the endometrium. So as the process of implantation was occurring, that digestive process of kind of breaking down the maternal tissue in order to get inside there and make a connection was the way that the embryo was getting nutrients. But that is only going to last for so long. So the developing embryo is going to need some more advanced nutrients in order to develop the correct way. And so we're going to have to get a placenta that forms. Placentation is the process with which the placenta forms, and this is a temporary organ. This is going to exchange nutrients and waste between the fetus and mother. So this is a formation, this is a union of the two tissue, the maternal and the fetal tissue. So a placental barrier is going to form, and that's going to keep the mother's blood from interacting with the fetal blood. Now, hopefully at this point, particularly since you have been through the immune system, that makes sense, right? We don't want mom's blood to interact with this new organism because the chances that the immune system will recognize this organism as different and possibly attack it are pretty high. Remember, this organism has a different set of DNA. It may be very similar to mom's DNA, but still it could be different. It could have a different blood type. It could have a lot of different factors inside of it. So mom's blood has to be kept separate from the fetal blood, and that's one of the things that the placenta is going to do. So you're going to get this formation of an organ that delivers nutrients, takes away waste, but it is not mom's blood going in. It's kind of an exchange region that goes between the two, and that's what the umbilical cord is. So the umbilical cord forms this connection with the placenta, and from the umbilical cord we get umbilical arteries. Now, the umbilical arteries carry deoxygenated blood away from the fetus. And I know this is another one of these things that could get confusing because normally when we think of arteries, we think arteries take oxygenated blood somewhere. But with the umbilical arteries, they are carrying deoxygenated blood away from the fetus. And it's the umbilical veins that carry deoxygenated blood and nutrients from the placenta. So the system of arteries and veins here is kind of like what you came to expect with the pulmonary system, where it was pulmonary arteries that were deoxygenated, pulmonary veins that were oxygenated. Well, it's the same with umbilical. So the process kind of flipped from what we usually understand of those two.